There we go. Okay, so we're going to talk about research tools. I'm really lucky to have Matthew Simon here. He is phenotyping a patient-derived Syngap related disorder model, and that's going to be, I think, I can't wait for his talk. But first, you're going to hear me talk about one specific thing that Syngap Research Fund is really dedicated to. And what I'm going to show you today is that SRF is making it easier to study SYNGAP1 related disorder with patient-relevant research tools. So you might be asking yourself, why? Why do we need that? And you might be asking yourself, what are these tools you're talking about? You might be asking, how do I find them? And you might be asking, what else? So I'm going to show you that today. First, I'll give you a little context. What we have available to us as a patient advocacy group is de-identified data and variant information. And what we can help make are cell models. And so we are starting with the assumption that SYNGAP1 related disorder is a haploinsufficiency, it's dominant, it's 100% penetrant, it's severe, and one of the hallmarks of its severity is the homozygote is non-viable. So as a basic scientist, the switch to thinking about the data per variant is a different way for me to think about the information. And so I just want to go through, and this is partly because of de-identified data. So I just want to go through what might we learn? What might we learn by having me show you what data and information is associated with each of the genetic variants that cause SYNGAP-related disorder in an actual patient. So what might we learn? The two big things, one is other disease models, and the other is genotype-phenotype correlations. So you know that Syngap is a haploinsufficiency. That means the disease model, it's thought to be a null, and so you have 50%. So potentially there are, instead of null, it could work less, it could work more, it could work in a new way, or it could be a spoiler, right? Those are the other kind of disease models. And then as far as genotype-phenotype correlations, one obvious example is isoforms. So if a variant affects one isoform over another and a different variant affects all isoforms, you might see a difference there, right? And then I just want you to note that the information I'm going to show you today, it's either public or it's ours to share or it's shared with permission. And the work that I'm going to show you is a work in progress, and it always will be. Okay, so why? Why do we need this kind of information? So Syngap is found by genetic report. It has a de novo pathogenic variant on it. And if you look in the this cohort of citizen data, where n equals 192, you see many different variants, and they're distributed across the gene. So we have 130 variants there that each are represented by one patient. That represents 2/3 of the cohort there are singletons. There are another 17 variants that are found in 2 to 8 patients. So the last third is represented in a handful of patients. So the disease model then, in a different analysis, I can tell you that 82% of that 192 have a protein truncating variant or some other type of null, like a large deletion. That means we pretty much understand the disease model. It's expected to be haploinsufficiency. The other 15%, the other 18% we don't actually know. About 15% of the patients have a missense in this cohort, and about 3% have something else that we aren't exactly sure how to describe. Okay, so what am I talking about? I'm talking about figuring out what information do we have from patients per variant. So you see 3 colors on the slide. Purple, the purple column is all about citizen health data, which is electronic health records. The blue column is about biorepository samples, and then the green is about models of disease, whether it's iPSCs or a mouse. So the easiest way to look at this data is to go horizontally. So if you start with the green 12 that I circled there, you see we have 12 iPSC models, 2 of which have a mouse for them. And if you go to the left, 34 of them, of those 12, they also have biorepository samples and citizen health data, right? That's kind of how you read that. And so you kind of go down the column and you see that at the bottom we have 4 iPSC lines that don't have anything else associated with it, right? So if you're a researcher and you want to study Syngap 1, we'd like you to know what lines there are. I don't have this information right this second, but we're getting QC and QA data for the different lines. And we want you to know when there's blood bank samples and electronic health records associated with it. Okay, so I should have talked a little bit about the 3 mice at Jack's. The next speaker is going to talk about one of the new mouse models and about phenotyping that. It's super exciting. And the last mouse model that I just put up there at the bottom, this is a humanized mouse. This is not associated with any, it's wild type. It's got the wild type human Syngap gene in it. So of course it doesn't have any medical records or blood at combined brain, but it is a really great model for studying a lot of things, including a lot of different precision medicine that requires human-specific DNA interaction, right? So that's going to be the human Syngap gene. And so I think you all know this, but it's the whole gene. So the whole locus is replaced. So how do you find these things? The most complete way is to email me. I have a big spreadsheet. I was thinking, how am I ever going to show a spreadsheet at this conference? So I'm not showing you the spreadsheet. That's not going to happen. But email me. We can talk if you're interested in this kind of information. I've got 2 other interesting things to tell you about. One is the Syngap variant viewer, and I'm going to spend a few slides on that. And the other is the Syngap missense server, and that's how I'll end. So the Syngap 1 variant viewer is an integrative genomics viewer that has curated Syngap 1 variants. So this does not have ClinVar, this doesn't have a lot of things. This has the spreadsheet that I made that has other data associated with it. Okay, so this is not a complete way to look at all Syngap variants. And then, like other genome browsers, it has the chromosome across the top there. It has the gene is the lowest horizontal line there in blue is the gene with its 19 exons from this particular transcript, which is the major isoform. And like other viewers, you can add tracks, and you can, in this one, you're going to add tracks as variant types. So if you're only interested in protein truncating variants, you can just add those. If you're only interested in missense, you can just add that. The other thing you can do is you can filter it by whether it has an associated biorepository sample or not, whether it has an iPSC line, whether it has a mouse line. So where did this come from? This is incredible, right? I did not do this. This is from Dr. Jessica Kaufman. She's actually a Syngap aunt and sister to Emily Barnes, and Hadley is her niece. And she is the dean of the School of Science and Technology at Endicott. And so it's great to have a bioinformaticist in the family. And she has a grad student, Ahmad Bashara, who has a poster here today. So I hope you can go see here's Ahmad. And he's been just doing great work for us and with us and helping me incredibly. So I hope you can see his stuff. So in addition, I worked on this. So it's with Syngap Research Fund, and it's all volunteer. This isn't funded, it's just volunteer. And some of the data we'll be showing you is from Citizen Health and Combined Brain. And of course, there's a whole lot of iPSC lines. They're from a a large number of labs. This is not all of them, but when you get if you talk to me, you'll see more of that. Okay, so I'm going to just show you a few screenshots and what you see with this viewer. And what I've done here is I have added the major types of mutants. Okay, so you're seeing the whole gene, you're seeing all, it's 147 from Citizen plus 6 others. So whatever, 140, so what's that, 173. So 153, so 153 variants here. So that's not that easy to see. So I'm gonna, let's see. So if you look in this vertical line, exon 8, 9, and 10, if we just look there, that's what you see on this slide. So that's exon 8 is the longer one on the left, 9 and 10 are to the right of it. You see two gray intronic mutations on either side of that blue bar. That's the intronic at the beginning and the end of exon 10. Okay, so now you're oriented to where we are. We've got a series of frame shift, missense, nonsense variants here. And so let's say we wanted to see out of these 40, there's 40 samples here, 40 variants. So if you wanted to see which of these 40 variants have biorepository samples associated, you just check that box. It goes down to 11. And say you wanted to know cell lines also, you check that box, and now you have 5. Anytime that you click on one of these variants, you're going to get a box come up that says a whole bunch of stuff about it, including the number of patients that are in the Citizen cohort. And it's often 1, but this one happens to be 2. And we're gonna be adding in library, like basically publication information. So if there's a specific publication with this variant, that'll be in the box too. Okay, so that's it on that weighted view. I'm going to talk to you now about what else. So like, that was great. What else can you do? So we have researchers, we have a couple of researchers now doing great work on missense variants. And so Dr. Pekka Pastila in Finland at University of Turku is doing in silico work. And Dr. Michael Courtney, also there in Finland with him, is doing biology assays. And so they work really well together. They are doing a ton of collaboration. Their work informs each other. What I'm going to show you today is 2 things. In orange, we have Dr. Pastila's, we have him, and that's going to represent his Syngap missense server. And I think we've talked about that before. It's the Syngap missense server has the top 3 buttons there. There's a 3D viewer, and then in the red box there is a data. It's like a data table, spreadsheets, yay. And then there's a whole bunch of information on that website. This is a beast of a website. It does way more things than I can really handle or figure out. But if you're a structural biologist, it's going to be fascinating. And there's lots and lots of information on how to use it, how to get the most out of this website. Okay, so this figure here, we're going to look just at missense. And I'm starting with the same 3 colors, the same information that I did before. So we've got citizen health in purple. For that, I have 23 missense variants that do have citizen health data, 2 that don't. On this table, of course, lots of missense variants don't have citizen health data, but for this table, we've got 2. And then the number with the biorepository data and the number with the iPSC lines. So that looks just like you would think, where you can read across and see what's there. So what else do we have? We also have whether or not the Syngap missense server gives a prediction on what the consequences of that missense amino acid change. We also have, so that's in orange, that was the Pekka Pastila information. In yellow, we have from Dr. Michael Courtney's lab. He is looking at a bunch of missense variants in Syngap. And so he's had to make plasmids to either deliver for in vitro, to do in vitro stuff, or to deliver with an AAV. So he has plasmids for these. And so this tells you whether or not a plasmid exists. And then over in pink, we have publications, whether our great graduate student volunteer, Richard Coca, who's at Boston University, he's a grad student at Boston University. Doesn't have Syngap in his family. He's just out of the goodness of his heart. He's just volunteering for us and doing a lot of library work for us. So anyway, so that's what's on there. So now you see you have kind of a rich understanding of what is available for these different variants. Just to wrap up, I want to make sure to remind you all that Syngap Research Fund's goal is to accelerate the availability of safe, effective, and meaningful treatments. And Syngap Research Fund was founded very intentionally with the values of collaboration, transparency, and urgency. And I think that this, even though this is a small project, I think it really highlights collaboration. It 100% highlights transparency. And it's really all because we have this urgency. So we want to, not only do we want to introduce you all to each other and have you collaborate, we want to make sure that we know how to share the information and the assets, plasmids, antibodies, whatever. So you can imagine adding a lot more things to this if you have a isoform specific antibody, if you have primers for a certain variant, the sky's the limit, really. If you have some kind of research asset or information or assay test that works on certain variants, and you're willing to share them, this is what we want. So the help we want from you, what information and reagents can you share to fill this out and make it an even more helpful resource for the Syngap One community? And then finally, even if you don't have something to share, I'd love to know, what did we forget? So this work has been, is really my boss, Mike, saying, where is that spreadsheet, Kathryn? Where's the spreadsheet? And so in making it, I realized how I was like, oh, but we could put this in. Oh, we could put this in. And so it became something more than a spreadsheet. It became a real love. And I hope it's useful for people.